Yes, we can. Yeah. Cool.
Okay, welcome all for this course. So before starting, I need to sure that everyone has created account on Orchestra account using the link on the chat. Also, if you need to search about this course on the website, you can check about this introduction to matrix factorizations and principal component analysis. So soon we'll launch the course once the speaker has joined. The speaker of today has joined. Just let me introduce her. The speaker of today is Dr. Aidens. She is a senior research scientist and biostatistician and also computation biologist at Dan-Farber Cancer Institute. She also works as senior research scientist at Harvard School of Public Health. Her research is focused on developing methods that leverage the growing of public genomic resources from several and different large-scale genomic projects. Dr. Aiden and her group has developed several methods to integrate genomic data, including DNA, RNA, and proteomic. She has also developed a biclustering method to analyze gene set enrichment. Also, she is a lead of Gene-Seq databases, and as part of that project, she created several R packages for integrating data analysis of multiple genomic datasets. She also a co-chair of the Bioconductor Community Advisory Report. Also, I encourage the attendees to join this group as they do several work for bioconductor community events. So, Dr. Aiden, is it for you now?
Thank you. Just let me get my video started.
Okay.
Okay. Thank you very, very much for the invite. I'm very pleased to be here. I've actually recently moved, so my affiliation, I've just moved from Boston. I've moved to Europe, so I'm back in Ireland at the University of Limerick. So I have slides and I have a workshop, and I would very much like this to be as interactive as possible. So please do post questions in the chat, raise your hand if you've got questions. I'm trying to see how many people we have on. I think there's enough people that this, Yeah, there's about 75 people. So yes, please do make this as interactive as possible. I know it's tough over Zoom to be interactive, but please do feel free to stop to ask questions, to either come on or to pose questions. Okay, so I've got a few slides and these are on Google Slides. I'm happy to share the link. Let me just post it on the chat. I'm just posting the link to these slides if you want to see them. And I think closed captions are working for this if people want to put closed captions on as well, if English isn't their first language. Okay. Can people see the slides?
Yes.
Okay, cool. So we will be running this workshop today. The green box has moved. Can you still see the slides?
Yes, we still can see it, yeah.
Okay. So we'll be running this workshop today in the Orchestra app. So the Orchestra is a platform provided by Sean Davis from Bioconductor, and it allows us to run an instance of our studio in the cloud. If you haven't used that before, just click on that and it will start up an instance. It can take a few minutes to start up, so you can just have that starting in the background. The workshop we'll be using is on GitHub, and this is the web link here, Pca Workshop. Okay, and I'll describe how to launch the instance. If you are launching the Orchestra instance in the background, your username and password is RStudio. The link that you will get, the web link that you will get, will be unique to you. So don't copy my web link, because if you do, you actually shut down mine, and so please do keep launch your own instance and don't copy the URL from somebody else. And the username is RStudio and the password is RStudio. Okay, and that's all that is. Okay, so within this workshop, I've got 4 different short vignettes, and this is really a beginner's guide to what is dimension reduction, through to presenting a couple of the more common versions that are used. I'm not really going to go into Tisney or UMAP. I can if people really want, but I haven't that prepared. I have presented on that in the past, but this was really trying to get the basics of principal components analysis. What actually is it doing? And there's 4 different vignettes on this. A really, really short example using wine, different wines, and tasting of wines as an example. There's then there's 2 other vignettes in this. One is on different functions that call principal components analysis. And even if you use some really like newer packages, you'll find that they are still calling many of these old functions if you look into the code. So it's really going down to the nuts and bolts. How actually is this working? And then I'm talking about correspondence analysis. In the field of single cell RNA-seq, there's a growing realization that these are count data, and count data need to be analyzed slightly differently. Principal component analysis was really for continuous data, and count data do better if you're decomposing the Pearson residuals, which is correspondence analysis. And then I've got an example of single cell RNA-seq. If people could do like an emoji thing, or how many people are using are doing single cell and how many people are doing other data types. If you do single cell analysis, could you just like put up an emoji? What are the different types of emoji? Put up a raised hand, so I just kind of know how many people are doing single cell. Jakub, can you actually detect how many people are?
But no raise hand yet.
So is there anybody? Okay. Well, then we may not. I'll give a call if people want to do it, or maybe just post in the chat. If people are interested in single cell, we can have a look at that. Okay, so so people are posting it in the chat. Okay, cool. So 1, 2, 3 people are using single cell. That's 4. Okay, Fatima as well. 5. Okay, we've got 5 people using single cell. If people want to post the data types that they're using, maybe we could have a quick count of that in the chat. Okay, so then I'll have a better idea of where to actually define this. So dimension reduction, principal component analysis, also called matrix factorization, latent variable decomposition, are methods where you're trying to take a very large data set and try to synthesize what are the main patterns in that data set. So you can think of it in terms of linear regression. In actually the first case described in 1904, it was actually described in that circumstance. Pearson described it as a linear regression. Or you can think of it as a way of pulling out all those variables that are correlated and actually synthesizing them into one component, and then pulling out the next set of variables that are correlated or that share traits and explaining them by one component. So essentially you're finding linear vectors through the data that explain most of the information. And in classical methods, the components are ranked such that the first component represents the most information in the data. The second component is orthogonal or uncorrelated to that, and that explains the next most amount of information in the data. So if you have a really simple experiment of cells that have just been treated with one compound, you might expect that the first component would represent the compound effect. The second component may represent some technical artifact or some other effect that's only represented by a subset of the cells. However, if you have a more complex data set where you've maybe human tissue and there's a lot of different cell types, you may need many, many more components in order to represent the variance in the data. Okay. Yes, if people are doing GWAS. Okay, so principal component analysis has been used a lot in GWAS and is actually typically quite often used to represent, to stratify GWAS data by major groups, such as by geography. Blood Rna-Seq, microbiome data. Microbiome data be count data depending on how it is. So some of the count-based information I'll represent with the count. I'm looking here at what data types people are using. So what is singular value decomposition? Well, singular value decomposition is just a matrix equation. It's a very simple equation where a given matrix X, we decompose it to 3 matrices. Okay, and these are the U, the D, and the V matrix here. The D matrix is just a diagonal matrix. So that means that everything in this matrix is 0 except for the diagonal. And the diagonal here represents how much information is captured by each component. Then we have the V and the D, or the left and the right singular values, represent here the weights of the columns or the weights of the rows in the new space. Okay, so for example, if we have a lot of information that's captured here in this first component, this is the first component in the U space, this is the first component in the V space, and this is the weight of that component in the new space. The number of components, which will here be K.
Hello, ID.
Yeah.
We cannot see your slide.
You can't see the slide. Okay, right.
Oh,
I did think. So what can you actually see?
First, we just see this Bio 21 Pca Workshop.
Okay, let's see. Right, share. I'll tell you what, I'll move this to the desktop and I'll share the entire desktop. That way I know for definite the entire thing is.
Yes, I think now it's working.
Can you see it now?
Yes, I can see now, yeah.
Okay, right. I'm not too sure. So you can see Matrix, many use singular value decomposition?
Yes.
Okay, cool. I wonder what was, okay. Oh, the joys of the joys of these virtual meetings. Okay, so sorry here. So let me take back a bit. So did you see this slide?
Yes, we can see now, yeah.
Okay, right. Okay, so the vignette is I'll just track for 5 minutes. Just sorry there, I'm not too sure what people captured and what was being broadcast. So there's various different vignettes in this. I will go through those. Matrix factorization here, or dimension reduction, you're trying to represent the data by new linear components. And so if this is the first axis, it will capture most of the information in the data, and this is the second axis, so the second linear vector, it will capture the next amount of information in the data. And how we calculate these is by singular value decomposition. We can also calculate these using eigenanalysis. We can also calculate these using linear regression. We can also calculate these using many different other mathematical frameworks, but SVD, or singular value decomposition, is the one that's used most frequently. And so if you do an SVD decomposition of a data, what you find is that the first you extract a number of, you can explain the new data set, the data set that you have, by a number of components K. The number K will be determined by the dimensionality of your existing data. It is either the number of rows N, the number of columns P minus 1, whichever is smaller. So if you have a matrix that is, 10,000 by 5, you can always explain that in 4 components. Okay. Or if you have a matrix that's 30,000 by 10, you could explain that in 9 components, obviously. So K here is the number of components. You will have the diagonal matrix here, where it's going to be a K matrix. Everything in this matrix is 0, except for the diagonal. And the diagonal here represents the amount of information captured on each component, where the term here at the 1, 1 position will represent that the amount of information captured on the first component. The V matrix here is the number of columns in the original matrix, and the first row here represents the first component. And equally here in the U matrix, the first column represents the first component, the first principal component. So let's have a look at that. So for example, this is this short vignette. This is the first vignette that's given. And this is a really, really simple data set, which has just got 5 rows and 4 columns. And it's 5 wines that were scored by 200 judges. And these are the number of judges that scored those wines according to a criteria of excellent, good, mediocre, or boring. So simply looking at this, we can see that these Grand Cru and Cru wines got very good scores, and we can see that some of these other wines didn't get good scores. So we could literally eyeball this matrix and actually work out what we're expecting to see. And I specifically picked something that was very simple. In France, the cru wines tend to have more restrictions legally. They tend to be more expensive. They tend to be better wines. The table wines, the Vinda table here, or the market wines here, tend to be like lower restrictions and tend to lower costs. So if we decompose this matrix, do I have the decomposition here? Oh, I didn't. And that is, so I can actually go to this. Let me just go to this actually and show you what this looks like. I'm just going to open up, oops. So can you see the Pca Workshop now?
Yes, we can see it.
So this basically is this vignette. And so it takes this data set here, okay? And what we do is we do a principal component analysis on it. Now, I have this little tangent thing here, which you can completely feel free to ignore. I was feeling a little bit like I wanted to stylize this. So I did a really fancy bar plot of the data using this very nice ggplot theme, but that is just the code to generate this fancy plot. So when you run the principal component analysis, the first step is to scale the data, okay? Now this is done inherently anytime you call Pca, but what happens, the steps that happen in a Pca is the first thing is we scale the data. So we center the data and we scale it. And so this here is that are those count data centered and scaled. And so if we look here at the excellent wines, we can see very quickly that the cru classic gets a very high score, followed by this other cru wine, followed by the Bordeaux, and the table wines here have negative scores. Okay, and equally, it's the exact opposite here with the boring wines. We can see this by looking at it. If we run an SVD, we have a 5x4 matrix, and these are the scores in the SVD space. Okay, we can see here positive scores. Sorry, this is the scale data, and then this is the data in the SVD space. So here is the diagonal matrix representing how much information was captured by each. And we can see that the first component captures here 3.5, so way more. And if we look at the percentages here, so we actually just square this and multiply by 100 to get percentage, we see 75% of the variance is captured by the first axis. This here is after we're running SVD. So this here is the SVD matrix here. Okay. And then if we look at the eigenvalues associated with this, and I'm going to show you the plots in a second, I just wanted to show you the data. We see here, if we actually look at the U matrix, we see here we have our 4 rows. And if we look at the weights here, so we have 4 factors or four components here. And on the first component, X1 here, we're seeing that our cru wines are on the negative end of the axis and the table wines are on the positive axis. And if we look here at the actual columns, excellent, good, mediocre, we've got negative scores here and we've got positive scores here. And if we actually plot that, we can see here that our excellent wines here on the negative end of the axis and the mediocre and the boring on the positive side. When we're looking at a principal component analysis, we are not worried about whether things get negative scores or positive scores. You can flip those axes. You're looking at the distinction from the origin, and the higher the weight from the origin and in the same direction from the origin. So we can always, if we don't like to see something on the negative or the positive end, you can just multiply by -1 and flip the axis around. That's the axes can be flipped either way. That is not important. We're just looking in the direction and the weights, so that the separation of the variables in that space. So just to summarize here, what principal components analysis is we're just scaling the data, we're running singular value decomposition, and then that is giving us a series of 3 matrices, which give us the weights of the rows in the space, the diagonal matrix, which are the eigenvalues, and the columns in the space. And then we can visualize those in our new space. Okay. The reason this works is because these matrices actually have some really, really nice properties. The U and the V, those loading matrices, are actually orthogonal, which means that they're uncorrelated, but also the product of their transpose gives you the identity matrix. Okay, so if we take U, we multiply it by the transpose of it by itself, we get the identity matrix. And the identity matrix is everything is 0 except the diagonals are 1. Okay, and equally, if we multiply the V matrix, so the row matrix, by its transpose, we also get the identity matrix. So these are really, really nice properties, and that is why this is so, this gives you a determined result. So you get the same result every single time you run it. Okay, so what I'd like you to do is go into the orchestra platform. Now, I've already started it here. And if you go into the orchestra platform, you'll be given the option here to choose a workshop. Now, it's actually sometimes difficult to find, because we now have like over 75 different workshops on this. But if you actually put in DIM, you'll get dimension reduction for beginners, and that's the newer version that I have actually done. That was in July this year. So if you click on that and you click on launch, I thought I'd already started this. Oh no, I had just logged in. So I didn't take my own advice. I didn't start this early enough. So mine is taking a second to start up. I'm going to just check the chat. Okay, so the password is just RStudio, and the username is RStudio. And that will start up an instance of RStudio in the cloud. So I presume people are familiar with RStudio. If people aren't, can I just get an idea? Do I need to introduce RStudio? Has there been other courses in RStudio that you've attended? Or are people familiar with R, or are they not familiar with R? If people could just post in the chat if you're familiar or not familiar, then I'll have a better idea. Just going to change the preferences here while you're doing that. Just going to make the font a little bit bigger, and every single time I can remember where it is. It's under tools. Okay, so familiar. Yes. Okay, so most people are familiar, so I don't need. Okay, right, cool. Okay, So within the RStudio instance, there's actually a lot of packages that are preloaded. Okay, so if we look here on this is your files, your plots, your packages here. Anything you do on this will be saved to the cloud instance. If you want to, you can, I'll show you maybe at the end if you want to be able to export things out of this. Okay, and the Pca Workshop here is actually called Pca Workshop. There's two ways to load this in. Either you can do load Pca Workshop, Pca, if I could spell, I'd be dangerous. Pca, or you can just click on it here. The easier way is just to click on it there. Okay, and that will actually load it in. I couldn't type, I think. Okay, so library Pca Workshop's what I meant to say. Library Pca Workshop. Load is for data sets. Okay, if I click on the actual tick box here, that will actually load the library. If I click on Pca Workshop here, you can click on the vignettes, and that will actually allow you to open the R source code or the HTML version on each of those. So there's R code associated with each of them. Sorry, if you click on source, it will give you what I actually wrote. And then if you click on the HTML, it'll bring up the HTML of that. Okay, so I explained a little bit about this this wine data set here. What I'd like you to do is I'd like you to bring in this data set, and I'd just like you to actually verify yourself that if you run an SVD and if you multiply the transpose of the U matrix and the V matrix by itself, that you get the identity matrix. Okay, my RStudio session disconnected. I'm going to reconnect that. In the meantime, can you actually do that? So let me just go back to the slides. And I'm going to give you a second, and I'm just going to catch up on the chat. Okay, here. So if you haven't loaded in the data and you're just in an ordinary R session, just make some fake data, run an SVD on it, and please verify that that you can actually do this multiplication. Okay, when you have that done, please post the code into the chat window as a verification that you got it working. Yes, you can interject with questions at any point in time. Oh, somebody has their hands up. Thank you for telling me that. I actually can't see.
Sorry, Aiden. So there was a participant who had their hand up, Nesreen. They did ask a question earlier, so perhaps Nesreen, if you want to go ahead, but I'm just quickly finding the question in the chat box. They just asked if you could please explain a little bit more around SVD. They didn't understand it very well when you did it the first time.
Okay, I think I saw the question, and then I realized that I think that was posted before the slides were being shown. So I explained it a second time. So did she understand it more, or do I need to go into it a little bit more?
Maybe a recap.
Sorry, Francis, go ahead.
Something, maybe a recap of the SVD method will be okay.
Yes, she says yes, thank you. Okay. Oh, don't try and do the pomological thing. That actually does have lots of problems, Harry. That was very, so let me show you what Harry posted a question there. Let me just show you what I'm talking about. So in this vignette, I actually have code to generate this image here, the pomological one, and it's this. It is actually problematic. It's not the easiest code. If you actually look at what I have there, I will have a val equals false on this, and that's because it actually tends to be pretty. The scale fill pomological thing does tend to be problematic. It requires a couple of things. All it does is generate this fancy image. It is not important at all. I was when I was actually writing this vignette, I kind of thought it might be nice to have, like, grapes and vineyard things, and pomological is kind of apples and vineyards, and it's just a theme that tends to be used in old school farming manuals on fruits, and that was just this color scheme. So this this paint pomological and thing, please don't even try to do this. This is how I generated this, but it actually tends to be this scale fill pomological just tends to be this package isn't the best, but I just wanted to include the code of how I actually made the figure. So don't try and do this part here, please. Just probably load in the data Bordeaux here, and then jump straight to here where you can just do the scale and just run SVD. Okay. So the code, I'm just putting up the code here on top, so you should be able to see what I've run and the history that I've run. So I've just loaded this package AD4, which has the Bordeaux data set. I've run scaling on the data set just to make it centered and true to scale and center the data, and then I've just run SVD. Okay, when you run SVD, you get these three resulting, the diagonal matrix, or just the vector of numbers here. You get the left and the right singular values. Okay, now just to prove that SVD is simply just these three matrices, X is equal to V U by D by the transpose of V, I can actually multiply here this U matrix. Oh my dear, this keeps jumping. I'm not too sure why. Okay, by the diagonal here of the D matrix, by the transpose of the V matrix, and you will see that this just regenerates my scaled matrix that I actually did the SVD on. Okay, so these three matrices, the product of them actually give me this. Okay. And here I'm just showing that the length is equal to the dimension of this, that basically all these dimensions equal up. What this little bit here, diagonal $D does, is it just gives you that diagonal matrix that I was talking about here. It gives me the 4x4 matrix, so I can do this matrix multiplication. So everything else is 0 except for the vector in the middle. If I want to see the percentage of variance captured on each component, I'm taking the square of these values in D here, dividing by the sum. And then in this case here, all I'm just doing is I'm rounding them up. So the 76%, 18%. So did anybody manage to multiply? I was expecting people to post code here. Did anybody manage to prove that the U times multiplied by the transpose of U is giving you the identity matrix, or V by its identity by its transpose gives you the identity matrix? I'm just kind of waiting to see if people actually managed that bit. Please post code if you actually manage it. Thank you, Godfrey. I'll give people another 3 minutes maybe. Okay, I think we should actually just move on. So let me just show you that. So if we take the U matrix here, we transpose it, we multiply it back by ourselves. Oops, sorry, I have S. We get this identity matrix. This is kind of, might be because it's got lots of kind of things that are almost 0, so I'm just rounding it. So this is the U matrix here. And equally, we can do this with the V values as well. Okay, we take the transpose here of the V values. Okay. So that's a specific property of SVD, is these, you've got the diagonal matrix, which is the weight of each of the components in the space. And then you have the left and the right singular values. And these have that unique property that you can actually multiply them by their transpose, and it gives you the identity matrix. And that's what enables this computation. Okay. Now, we've just done this kind of lengthy, where we actually took the data, we scaled it, we ran SVD manually. That in practice, that's actually not what you're going to do. You will actually call a Pca function in R. And so you'll probably call precomp or princomp or one of the other functions. And so princomp here is one of the ones that's most widely used. Okay. And if I do precomp for running principal component analysis, let me just bring up the help here, you can see it. It performs principal components on a given matrix and returns the result as an object of precomp class. Okay. And here, if I look at the, just go back to the help there, where it was. Okay. This here, you can run this with scale equals true or not scale equals true. Okay. By default, within this, if we want to see the code. Let me just see. Let me just get methods here to actually bring up the code of this. This is an S3 function. And so I can just type methods, and I see that there's two different versions, .default or .formula. So I can actually just bring that up. It's not finding it, but I know it's part of stats. It mustn't be exported by the namespace. So if I just do stats and 3 colons, that will allow me to see it. That's kind of a trick. If you want to see code and you can't see it, just type methods. If it's an S3 function, it will tell you that there might be more than one type of it. So depending on what the data type is, it does something slightly different. So in case if it gets what is called default or formula, it does something different. And then it's saying here that it's not been, can't see it. So if you do name of the package 3 colons, you can see the function then. So by default, the first thing it's doing is it's running scale. Okay. And by default, center is equal to true and scale is equal to false. Note that. I'll tell you a little bit about that in a second, and then we know it's running SVD. Okay, so this is the way that most of principal component analysis is calculated, is that you're running a function like precomp, but behind the scenes, what they're doing is they're doing a scaling, and then they're running SVD. And I just wanted to show that. Okay, so within precomp here, okay, we get a number of different, if I look at the structure of what it returns, str, we give you the structure of what it's returning here. We see that the first thing here is the standard deviation. So this is our D matrix. We're getting the rotation matrix. We're getting the centered and the scaled data. Okay, so let's have a look at some of this. So the first thing here is I wanted to see this precomp $X here. This is my actual scores on PC1, PC2 for the actual wines. And then we can also look at the rotation. And that actually gives us the scores on PC1 and PC2 of the actual columns. Now you might notice that when we actually looked at S, these values here are not the exact, they're the same here for these columns. The V matrix here is the same as this rotation matrix. But we're kind of going, but wait a second, why are our wines not the exact same? What's the difference here? And that is because these are the actual scores, not just the actual weights. So how we actually go from one to the other here is we actually just, you'll multiply by the actual, the diagonal matrix. And if we multiply by the diagonal matrix here, we'll see that that's the exact same. And these then are the actual scores in the space. Now, one of the things that got me confused when I started looking at sort of different versions of principal component analysis and how it's implemented, it's very, very simple mathematically, but the way it was implemented, it's kind of different depending on the different package. And that's actually why I wrote the other vignette in this space, this principal component analysis one. And what, sorry, that's not this one. I'll go back to the packages. Let me just show you actually on the website, maybe that might be less confusing. On the actual website here, there's actually multiple ones. And the second one here is this principal component analysis in R. And with that, what I actually do is I go through different packages that run SVD and that run Pca, and I say how they all relate to each other. Okay, because it's very easy to run one and not realize that the defaults in one are slightly different to the defaults in the other, or working out what the differences between the weights and the scores. And so this here is kind of my translation here of what we're looking at. What are the loadings? What are the scores in each of them? And basically different ways to plot it. So I have a toy data set here. And I show you, actually I also show how to compute principal component analysis using eigenanalysis instead of SVD. I then run pre-comp. I run correlation based Pca. I'm going to explain that in a second. I run the scale equals true in pre-comp here. I run print-comp, which is another version. I use Pca in Factominer, and I also use another package AD4. And in each case, I show that they're all generating the same result and what the relationship between them all are. And I compare all of these different methods. And I show here that in every single case, if we're looking at the eigenvalues, we're actually getting the same result, even though they're actually doing slightly different things. Okay. So that was one of the things that I have in that. Let me just go back. If people have questions, please post them. Okay. So let's, so we've done this here. We've shown here that orthogonality. So let's talk a little bit more about the results and how to interpret them. Okay. So as I mentioned, when you run principal component analysis using SVD, you get the left and the right singular values, which correspond to the weights of the rows and the columns in the new space, and you also get this diagonal matrix D. You can compute this via linear regression, eigenanalysis, singular value decomposition, latent factor analysis. There's many, many different ways. And in that second vignette that I describe, I show how to use eigenanalysis and SVD. One of the most popular, one of the most widely used linear regression methods is a method called NIPALS, N-I-P-A-L-S. And then these vectors that you get back, this like these new, the U and the V matrix can be called your principal components, principal axis, latent vector. There's many, many different ways that these are referred to, depending on what methods are used to compute these values. But at the end of the day, you're actually taking your matrix and you're still generating out the same information. Okay, it's just the method that you use to compute it means that we're actually using different terminology. And I think people new to the area sometimes find all this myriad of different terminology for the exact same thing to be quite confusing. So that's why I've actually mentioned these here. But this diagonal matrix, where you can actually see the percentage, remember we had 74% of the variance was represented on principal component 1 in the wine matrix. How do you decide how many components to look at? We had a matrix there with 4 PC1, PC2, PC3, PC4. Do we look at all 4? Do we just look at the first 2? What do we decide to do? And this is an unsupervised exploratory method. So there's no right or wrong here. If you want to look at PC1 and PC3 or PC2 and PC4, you can. Each one should be telling you something different about the data and something new about that data. And whether that's something of biological significance or a technical artifact, you have to work that out. One way people do use, and this is a quick, rule of thumb rather than anything else, is if you look at the eigenvalues and if you plot them, you can actually see a drop. And these plots are called a scree plot. And a scree is actually, if you're looking at a mountainside, where you see the kind of loose gravel at the bottom of the mountain, where you have that steep drop and then you have the moraine, that there is basically where you could kind of call the elbow, or sometimes these are called elbow plots as well. And so as you kind of come down to that elbow, that's kind of where you make the cutoff. And then at that point in time, you can say, well, actually, in this case here, I'm probably not interested in all of these components here that add very little information. I'm going to look at these main components, as these are likely to have most of the information in the data that I'm interested in. And so this here is called the Kaiser rule, which is basically, you're just eyeballing that elbow plot and looking at the ones above the elbow. You'll see in a lot of machine learning approaches, it's actually just done as an arbitrary number. So people will look at the first 10 components or the first 30 components in an analysis, and that they rarely do they actually consider the number of components. Within this slide deck, I have other permutation-based approaches that you can use as well. And those are actually very effective if you want to work out how many components are useful. Other people do, a rough cutoff, like how many components are required to get 75% of the variance or 80% of the variance. So there's no hard and fast rule here. Some methods are more rigorous than others, but this is an exploratory data analysis. Okay. So I mentioned again that this second component looks at different methods that we can use to basically generate the exact same data. And all of these different methods tend to have different approaches in what they actually call the output. And that's the reason why I actually generate these tables. So it's actually easier if you're using somebody else's code to work out what they're actually looking at. In all cases, as I mentioned, there's many, many different approaches here. But what we're trying to do is just find some order in the data. And so sometimes this is called ordination. What you're trying to find is, we'll say that axis 1 represents 5 cars here, axis 2 represents 4 trucks, and axis 3 represents 2 utility vehicles. You're trying to find patterns in the data. Okay. One thing that's important to note is that there's 2 principal forms of Pca. And one is called covariance-based Pca, and the other is called correlation-based Pca. But the only difference between these is in that initial step. So you're still running SVD, but remember we ran the data, we both centered and scaled the data. If you only centered the data and then run SVD, that's actually covariance based. If you center and scale, then it's correlation based. That is the simplest way to understand the difference between these. Now, we wrote, there was this now is, I suppose this is 2019, but there was a paper that was from Sun et al, and they looked at many, many different dimension reduction approaches for analyzing single cell data. They looked at factor analysis, principal component analysis, non-negative matrix factorization, and then a lot of newer methods that have been described. In this analysis, mostly because of scalability, speed, consistency, and success rate, they actually ranked factor analysis and principal component analysis as two of the methods that performed better than anything else. Here, blue was good, orange was intermediate, and gray here were poor. But when I actually went back to reproduce this and I just ran Pca, I actually had to go back and check their counts, because what they actually did here is they took their normalized counts, they centered, and they didn't scale. So they actually ran covariance-based Pca when they actually did this here, because the scale is equal to false. So that is the difference. And of course, I would never have known this unless they provided their code. So it's actually very, very good that they did. But just to note that there is two different versions here, and it depends on whether you center and scale the data. So we did a little mini review, and this is last year now, this is in 2020, where we looked at the effect of like pre-processing in your data and how that impacts principal component analysis. And so this here is like some single cell data. This is from Matt Ritchie's lab. This is the SC mixology data. And what they had was they had 3 different lung cancer cell lines. This is single cell Rna-Seq. And they ran these, they mixed them and they ran them on 3 different platforms, CellSeq, 10x, and DropSeq. Now this is a really, really, really simple data set. And so we looked at, well, let's actually have a look at this processing and how important it is. And so if you run the 3 platforms here are blue, orange, and green. So theoretically, there should be no difference on the platforms. These should all overlap, and we should see the differences in the 3 cell lines. Now, for simplicity's sake here, I've called the cell lines A, B, C, and A, B, and C. So this bottom left here, or this one here, is what we'd expect. We'd expect so the orange, the different colors to be overlaid on each other, and good separation of the 3 cell lines A, B, and C. Now, if we don't pre-process the data and we just do an SVD on the data, we see we actually don't separate the cell lines. Okay, and in fact, here on this first component, we've got an arch effect, which I'll describe what that is in a second. And you see here, if we even take the log counts on the data, we're still not getting any separation of the data. Because it's important to center the data, because then you have the values separated around 0 and you can get this decomposition. If we ran just SVD on the centered data, so we scale center equals true, scale equals false. So we're just centering around 0. So if we take each column in the data set and we subtract the mean, we're centering on the data. Okay. We see that if we take the raw counts here, we're still getting very poor separation. But if we use the log counts here, we are getting separation of our 3 groups. You're still seeing that there's quite a big difference in these platforms. We can see that the 10x are projected further out in space, more so than the CellSeq and DropSeq. But we see that these cell lines A, B, and C are all being separated. If we look at the correlation Base Pca, where basically we have both centered and scaled, so subtracting the mean and dividing by the standard deviation, we're actually seeing, so let me just go back here. So in this case, we're taking each value, we take the column mean away, and we divide by the scaling factor, which is normally the standard deviation. So we're centering the data, so the mean is now 0, and the standard deviation is 1, okay? We see that on the raw counts, we're not getting the separation. On the log counts, we are, okay? However, so just sort of summarizing this, centering is important and scaling is important. So we are seeing better separation here if we both scale and center, which is this correlation Pca. Now, I mentioned here that there's this arch effect. Let me explain what that is. So with Pca, you actually often see, if you've got gradients in the data, or if you've got correlations structure within the data, you see something called an arch effect. And let me describe what that is. This arch effect, you normally see that you have the data is pushed to one side of the origin, and you see this kind of in the second component, you see separation. You don't see separation on one component, and you see separation on the second component. So the data ends up looking like this big arch or this big horseshoe. Okay. This has been well described. There's many. Susan Holmes did a really good review of this and actually presented multiple different instances of it. This was a discussion, this is from this is 2008, and it was the first time I'd run principal component analysis on single cell RNA-seq, and the first thing I'm seeing here is like horseshoes all over the place. And then, you know, it was basically, it started this whole Twitter thread where Simona like actually commented, oh, I see that a lot in metabolomics data. And Watson was saying, well, we see it in human genetic variation data as well. And Jamie Morton here has actually written a really nice review of this in microbial data. And when you were saying here that you were looking at GWAS data and you're looking at microbiome data, I think it's important to recognize that these horseshoes exist in PCA. And the horseshoes exist because there's correlation structure, there's gradients within the data, and there's also a lot of zeros. And although we can do things like logging the data for single cell RNA-seq, it actually isn't the best approach. There's been multiple approaches, multiple papers now, the Towns paper, where they show that doing this log transforming of single cell RNA-seq, at least where there's a lot of zeros, you actually distort the data. So really we need to start looking at better approaches. Is scaling and centering the data sufficient in order to get a normal distribution? And if you look at single cell RNA-seq, we actually see that it's not sufficient. And I'll talk a little bit about that in a second. But this here is James Morton's review, which is a really nice one about microbial data. So this here, he's got a really nice one. He puts in non-zero values, and you can see this like really nice horseshoe, this artificial horseshoe, but he actually looks at various different approaches here for actually trying to overcome these issues of horseshoes in this data. So that's one thing to note about. Okay, so when do you use PCA? That's a great question asked by Sanwali. Sorry if I've just mispronounced your name, I'm sorry. So principal component analysis is actually best suited to data that's continuous and that's normally distributed. But there's actually many other forms of dimension reduction analysis that are better suited to data where we have lots of zeros and where we have gradients in the data. And so this is a review that we wrote, wow, 2016, 5 years ago now. And others, Susan Holmes has also written a really nice review on 10 tips for dimension reduction analysis. But there's many different approaches. And interestingly enough, where you actually look at these, if we look at principal component analysis, this is SVD of data that's been centered and scaled on the column. So you take each column in your data, you subtract the mean, you divide by the standard deviation. Okay. Correspondence analysis is a method that's been around since the 1960s, 1970s, and that's better designed for count data, and it's actually it's SVD on the Pearson residuals, and I'll show you how that's computed in a second. It's very simple. Non-symmetric correspondence analysis is an extension of correspondence analysis to deal a little bit with the gradients. In microbial analysis, in microbiome analysis, Pca has been used a lot, which is principal coordinate analysis. And that's actually SVD of a distance matrix. So you actually take, you can compute any distance you want. So you could do Euclidean distance, you could do correlation, you could do Manhattan distance, you can do a distance based on phylogenetic distances, It can be any kind of distance measure, but it's basically decomposition of a distance matrix. Okay. And then there's many other different dimension reduction approaches, which aren't these classical ones that use SVD, such as NMF or independent component analysis. And those basically have reduced constraints. So those don't require the axes to be orthogonal or uncorrelated to each other. And those can be very powerful if you want to find local effects in the data. So as I remember, I said, the SVD, you're trying to find a vector through the entire data set which represents variance. Well, if you've got a data set that's got a lot of complexity, sometimes you want to find local patterns. So something that's very strong in a small subset of the data. And so some of these other methods are better at dealing with kind of nonlinear effects and with the local variance. And then there's other methods where you can find global and local effects both in the same data in data sets. And then there's various different sparse implementations as well. In general, when you see a sparse implementation, what it's doing is it's doing a lasso regression, or it's doing something to remove features. So at its most basic, it's you do principal component analysis, you look at the first component, you take the features that have the highest weight in the first component, and you send all of the other features to 0. Or you send a lot of the eigenvalues to 0 and in that diagonal matrix. And once you send those to 0, you can then multiply your matrix back again and you can do the decomposition again, or you can just ignore lots of the data. But a lot of the times with sparse, what you're just trying to do is drop lots of the data from the analysis. Okay, so as I mentioned, so when we looked at when we looked here at this mixology data, I was saying that principal component analysis, because it's just scaling and centering the data and isn't really designed for count data, and single cell Rna-Seq are count data. And I was mentioned here that this method, correspondence analysis, is actually designed for count data. And when we actually apply correspondence analysis on the XE mixology data, we see way better results. So up here on the top left, we see principal component analysis. And although you're getting separation of the 3 cell lines, we can see very clearly the effect of the different platforms. So the 10x blue ones, the cell-seq orange ones, and the drop-seq green ones aren't actually, we're still having that technical effect here. We can see that the weights are very different. And that's partially due to library depth. We can scale and remove, we can do a normalization for the library depth, and that does help, but it doesn't remove it entirely. If we just apply correspondence analysis, we actually, whether we use the counts or the log counts, we actually get much better overlaying of those 3 platforms or alignment of those 3 platforms. We don't even need to use a specialized data alignment process because we're now using a better pre-processing of the data. We're looking at the Pearson residuals of the data rather than just on the scaled and centered data. Okay. And it doesn't make a difference whether we look at the counts or the log counts, because this logging, which does distort the data, isn't actually required. So sometimes just considering which implementation of dimension reduction analysis and what's most appropriate for the data is actually worthwhile, rather than simply throwing principal component analysis at everything. We actually have this paper, it's unpublished there. We actually have the paper ready and it should be up on bioRxiv today or tomorrow. We actually did a lot more on this rather than just correspondence analysis. Okay, we've implemented correspondence analysis for single cell Rna-Seq in the Corral package. Okay, so let's go back to where we were. Oh, I just got out of the slides. Am I still sharing? I am. Okay, so what we have, So we can do a little bit about visualization, plotting. I can go into the different types of implementation, so the different functions for calling principal component analysis. I can describe correspondence analysis, or I can do an example of scRNA-Seq. What would people like? Can you please just post in the chat kind of what you might like? And also if people have questions. Okay. Hi, I should know how to pronounce your name. Heidi, what does the function scale equals t and scale equals f mean? That scale equals true or scale equals false? Just answering your question here. So that is if you do, so centering is dividing by the mean and scaling is dividing by the standard deviation typically. How do I add the ellipses in Pca? I will show you that, Nicholas. So. Okay. RNA-Seq, single cell RNA-Seq, a bit of everything, single cell RNA-Seq, RNA-Seq. Which method is best to test for significance? Somwali, can you actually either come on and say what you mean by that, or can you, I don't actually know what you mean. I don't understand the question, sorry.
Hi, thank you. So my question is to ask for a test to determine if there's any statistical significance in the groups that you're looking at. So for instance, if you see in your page that you're sharing now, like if you're looking at the green and blue, would you say those are significantly different in terms of their grouping? Or would you say they are not statistically significant? Or is it just limited to pure visualization?
So Pca is pure visualization. And in fact, I can actually, so this here was looking at the iris data set. Let me just, I can just run through this. This is on which, I'll just do this for you if you want to see it, see that visualization. Okay, so this was in the eigenvalues. We've discussed about eigenvalues and eigenvectors. We've discussed about scores. Okay. Oh, so this was in the other vignette, was it? It was in the Pca vignette. Okay, let me just go back. So, well, there's two things I want to do here. Let me just show you two things. So if we do, maybe we do Pca Explorer because that's actually easy to do. So in this case here, you can see that there's 4 groups and they look separated. Now they can look separated maybe on PC1 and PC2, and then they may not look separated on PC3 or PC4. So then the question is, well, do you test over all 4 components, or you do test over individual components? Most of the time, if people are testing components, they test each component independently to see if it's associated with a factor, with some known factor, in this case here, condition or tissue. You would test principal component 1, then you would test principal component 2, you test principal component 3, principal component 4. And then you can actually do a plot to say, well, actually principal component 1, 2, 3, and 4 are associated with this, 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 and this. In fact, that's how some of the batch removal methods work. So surrogate variable analysis is actually just that. It's doing a PCA on the data and then trying to identify which component is associated with batch. And then if you find that component 3 is associated with batch, what you do is you send the third element on the D matrix to 0. Now you back multiply your data, and then you've removed that effect. Maybe I'm not explaining that really well. Let me just, so if you, actually we did that, didn't we? So did I do multiplication back again? I do explain it in this a little bit. Just going to copy and paste, easier. Here. So we know here that if we multiply the U, the D, and the V matrix back, we actually return the matrix as it is. If we take an element within this diagonal here and we want to remove that effect, we can actually just send this value here to 0. We can back multiply it, and then you're actually removing that effect from the data. Because what it will do is it will remove, when these matrices both multiply out, you're actually just going to take away that third component from the data. So that actually is used in some methods where you basically test each component for its association with something. So we could equally take principal component here and see if it's associated with excellence or with something else to see what is actually explaining. In a lot of cases, we may not know a priori what are the covariates that we're interested in, and we may not see that. We may not be able to test it. But yes, you can test each component here. You can just do an ANOVA or linear regression to actually see, is component 1 associated with a variable, is component 2, is component 3, is component 4. And then you can say, well, actually, this factor, I don't know, the price of the wine is explained by principal component 1 and 2 together, or just by principal component 1. Some people have used that as well as a method for looking at gene sets. So there's a couple of different gene set analysis methods that have used where you do decomposition and you then say, well, does this, if I take this subset of genes, I do a principal component on it, does it now separate out my 2 groups? If it separates out my 2 groups, I now say that this gene set, which is explained by principal component 1 here, is actually associated with my condition. So people do test these. There is also supervised principal component analysis. So if you have some sort of a classification that you want to impose on the decomposition in order to get the variables that are maximally separated in space, you can run a priori a supervised decomposition. And so if you have fewer features than you have cases, you can just run a discriminant analysis, which is essentially a between and within group principal component analysis. You can run a between group principal component analysis if you've got more features than you have cases, or there's various sparse implementations of principal component analysis that are supervised as well that you can apply. Okay, so I was going to do a little bit on visualization there. So at the bottom of this, I mentioned Federico Marini's package. they have a really nice package called Pca Explorer. So this is actually a really nice visualization here. In also in my other vignette, I kind of do it in a little bit more detail. I go back to my packages. Bring up the help. There's 3 vignettes in the Pca vignette. Okay. At the end, I described the different methods that all do the same thing. And at the end of this, I actually have a couple of different methods here for doing exploring a Pca. So let me just save out a principal component analysis. Okay. And I'm just going to run Pca Explorer as just one. There's many, many different approaches. Federico's package is really, really good as well. Let me just, and then at the bottom of this, I also have various different approaches here for like drawing with ggplot. There's also, maybe I'll just run this code here. So this here is the iris dataset, which is a simple dataset of flowers. So looking at the sepal and the petals, and looking at their width and their length for different species of flowers. And so here you can see that these flowers are very different on principal component 1. On principal component 2, it's quite difficult to actually, it is not the species here that's separating them out. And we may actually well decide, right, actually I want to look at other components here as well. And so, and in this case here, we've actually framed around them, but we can see the principal component analysis 3 isn't separating these out here. And so one of the functions that I kind of like, but you know, you can, this is a different way for drawing densities around the plots. If you want to see, I can just show you this one here, you can actually just see a density plot of where the points are. And I wanted to bring up Explorer. Where is it? It's various different ways. Explorer. And I'm going to do. This is another application that runs. Please let me see if you can tell this, because this is the shiny one. Can you see this? It's a shiny application with the eigenvalues. Yes, you can see.
Yes,
okay.
You can see.
So this is kind of a nice visualization. It's just a little shiny app. And this will allow you, if you've got something that's a little bit more complex. I do actually want to extend this so we can do this maybe for single cell Rna-Seq or whatever, but it'll allow me to say, right, I actually want to look at PC2 and PC4 or PC1 and PC4. It'll allow me to change the label size. You can do a lasso selection if you want to slew. You can export the R code and you can export it as an SVG, as an image. We can look at the variable data. We can look at the actual like genes in this case here. We can get it to show the labels if we want. This isn't great if you've got gene expression data. You definitely don't want to turn on all of the labels. But this will allow you to actually, and you can mouse over anything and it will tell you what the name of the gene is. So this is one shiny app. There are many. Oh, this was the eigenvalues here that I was mentioning before. That, I don't know if you can see here, this is eigenvalue 1, 2, 3, 4. This is the actual variance captured by each. So 73% was captured on the first component, 23% on the second. And then you can look at the cumulative variance. You can say, right, between number 1 and number 2, I'm getting over 90% of the data. So I'm happy just to look at number 1 and 2. But there's many, many, many different Shiny apps and different ways to visualize these. And this one is a nice one because you could actually just pull out the R code for it as well. I want to make sure that I'm shutting this down without shutting down my R session. I hope if I close this, I'm doing something right. No. Did I just shut down? Okay, right. I'm sorry. I tried to shut down my Shiny app and I've actually just shut down my entire Chrome window, which isn't good. I'm going to have to start up again. And did I, in my genius, remember my orchestra? I never copied my orchestra thing. Right. What was there? Isn't NMF recommended? Okay, so I'm just looking at the questions here. So I just did a little bit about adding ellipses. They're using ggplot. That's most plotting functions for Pca will allow you to add on ellipses. It depends on which function you use. But the code there for running it on ggplot was shown. NMF recommended for Rna-Seq. NMF is actually good for most data sets. There's pros and cons to NMF. So NMF is really good for finding local local attributes within the data set. It's additive, which is really nice. So the components are additive. But the disadvantage to NMF is it is incredibly slow. So it's an iterative algorithm. It's using gradient descent to actually find each of the components. I published a review a couple of years ago with Elena Fertig's group, and we describe NMF in quite a lot of detail in that. I like NMF, but I'm always hesitant to run it on a big data set because it just, it can take quite a while to run. That's my only caveat there. There are a lot of other kind of newer machine learning approaches like variational autoencoders. And if you set a variational autoencoder down to its bare basics, it's actually Pca. So there's a ton of different methods that are kind of built on this idea of finding the reduced dimensions, or basically finding these vectors in the data that explain the most of the information in the data. And NMF is very powerful. I don't think that takes away the importance of actually considering, you know, how the data is transformed before you do things. Okay. I have a question about single cell data. If I want to investigate multiple data sets from different single cell RNA-seq studies and platforms, would Pca be the right method to do visualization? No. Most people use Pca. If you use decomposition based on the Pearson residuals or on another method that's better for count data, you actually get much better performance. I'll do the single cell RNA-seq tutorial now, and so you will hopefully see what I'm talking about, and you'll see the improvements there. I am just restarting here. Okay. Okay, share screen again. Right. You should now see our studio again.
Yes.
Cool. Okay. So let me go through the Pca. So there's two different Pca ones that we have here. And so, there's two vignettes that are relevant for single cell RNA-seq. And so one here is just a description of correspondence analysis. And I wrote this one here, and it sort of shows why correspondence analysis is the same thing as decomposition of the Pearson residuals. And obviously, Pearson residuals is a good way to deal with Poisson data or count data. And I go into quite a lot of background about it. And there's a toy data set here. This here is a toy data set. And the idea here is this is count data that's simulated to look like single cell RNA-seq. This has been used by different publications. And there's 3 batches in it. There's there's 3 clusters and there's a batch effect. So the clusters here are the red, the green, and the blue. And obviously the clusters aren't being separated. Instead, what we're seeing on the first component is the batch effect. And this is when we run Pca on the data. If we now run, and we see here that if we actually look at this principal component analysis, we see that the actual main effect on the first component is batch. Okay. If we run, this is just showing Herbal as a faster approach for running principal component analysis. If we run correspondence analysis, which is the decomposition of the Pearson residuals, we actually get a very different result. So I'll show you the result first. And then, so here we actually get group A, B, and C are now actually separating out. We don't have the batch effect on the first component. How do we do this transformation and why is it different? Well, rather than just centering and scaling the data, what we're actually doing is we're decomposing a data that's essentially the observed minus the expected over the root of the expected, which is basically your Pearson chi-squared statistic. And the way that we actually calculate that is this really, really simple approach. So if we have a matrix here of count data where we've got a lot of zeros, what we can do is we can find the row weight, which is basically the sum of the row divided by the total sum of the data set. We can equally find the column weight, which is the sum of the column divided by the total sum of the data. So the total sum here is n dot dot. If we get the column weight here is the sum of basically this column. So this is, so for every single value in the data set, we can now say that its expected value would be the product of the row weight by the column weight. So our expected value here is actually just the product of the row and the column weight, which is basically the contribution of this row to the entire matrix, its sum over the entire sum of the contribution of the column to the entire matrix, which is its sum over the entire sum. Okay, so Rij here is the row weight by the column weight. So we basically take our actual value here, we actually subtract the expected value, the product of the row and column weight, and we divide by the root. So this is basically the transformation. And so essentially what they're considering, we're dual scaling the data. We're actually scaling on both the columns and the rows. And when you actually scale on both the columns and the rows, we actually get a different representation of the data that's now subjected to singular value decomposition. And now each value in this new matrix represents the strength of association between this column and this gene. And this then is very different values than when you actually do the decomposition, you're looking in a different space. Essentially, when you're doing a principal component space, you're just doing the scaled and centered data. And essentially what you're doing is you're looking at the maximum difference. So essentially more of a Euclidean distance in space. Whereas here you're looking at a codependence or an association space. So the association between genes and samples. Okay. And when, and I actually just compute this really, really simply. My row weight here is the row sums divided by the sum of the matrix. My column sums here is my column sums divided by the sum of the matrix. My expected value is then I'm just doing the product of these throughout the matrix. And then I actually get my value, and then I do an SVD in it. So I compute it like little bit by little bit, so you see exactly what we're doing. And then I visualize this with Explorer. This is scattered 3D, which is yet another visualization way. So in each of these little vignettes, I've included different visualization approaches. Just working. And then this approach, this correspondence analysis, which as you can see is just a really, really simple matrix computation that's determined, actually gives you the same result as GLM Pca, which is this generalized Pca approach that was described by Townsend Isari, which is a very good method, but they use it in an iterative way, and it's actually quite slow. Okay, and we implemented that in the Corral package. Okay, so let's actually just take some real single cell Rna-Seq and actually just do some examples. Okay, cool. So I'm just going to load some libraries here, and I'm just copying and pasting. So the first library that I'm loading is the single cell experiment library. And this is the main library in Bioconductor for representing single cell data. I'm loading the Corral library, which is the library that we wrote myself, and well, my student Lauren Shu wrote it. And it's for doing correspondence analysis, decomposition of single cell RNA-seq. We're loading ggplot2 for plotting, and we're loading UWAP, which actually does UMAP. It actually is a package for doing UMAP. And then we're loading a data package, Duo Clustering 2008, that Charles Soneson and others put together that contains several benchmarking data sets. Okay, and the data set that we're loading is this SCEs, or single cell experiment full ZingMix 4. And this data set is within the dual clustering package. And what it is the ZingMix 4 equal is 4 different cell types that were mixed in equal measures. So the ZingMix data set was PBMC blood cells that were purified out. And then there's the duo clustering will ask if you want to cache the data. So I'm just going yes here. Yes, please do it, so I don't have to keep downloading the data multiple times. So show you the ZingMix 4 data set, what it looks like. Oh, did not save it. Let me just do it again. It's a single cell experiment data set. It's got 15,000 genes and just under 4,000 cells. Okay. Within duo clustering, if you look at it, there's multiple different data sets available. And the ZingMix data sets, there's multiple ZingMix data sets. So there's 4 cell types that were mixed in equal measure, there's cell types that were mixed in unequal measure, and then there's a ZingMix 8, which was 8 cell types that were mixed. And so this and the SC mixology data are 2 kind of benchmarking data sets that have been widely used for single cell methods development. Okay, so let's have a look at this. Okay, so this is the standard single cell RNA-seq. We know it's single cell RNA-seq experiment. We have 3 different data types or assays within this. We have the counts, we have the log counts, and we have the normed counts. Our row names here are as ensemble gene IDs, so we know that ENSG is human, and these are just ensemble gene IDs. We also have row data here where we have gene symbols and other information about the genes. And the col names in single cell RNA-seq data sets tend to be really boring. They're always like cell 1, 2, 3, 4, 5. But in this case, what we do actually have is they're actually labeled a little bit better, so we know that there's B cells and regulatory T cells. Let me just show you, just do table. So the phen ID here is basically the cell types. In this case here, we've got 4 cell types. There's B cells, there's CD14 positive monocytes, there's naive cytotoxic cells, and T cells, and there's regulatory T cells. And you can see that they're pretty much equal. There's around 1,000 cells of each. Okay. So just for ease here, I'm just going to extract out the assays. You don't have to do this. And so if I do assay here, that allows me to extract out sort of the matrix. And in this case here, like counts mat is just the matrix here of B cells and T cells. I wouldn't advise this for bigger data sets, but this is quite a small data set, so it's actually very easy to do. Okay. Now, if we want to run a Pca on this, so the first thing here, we tend to do this. It's standard practice if you run some of the Surrogate pipelines, is to actually filter the most variable genes. And this, as within all of these vignettes, I try to bring it down to bare. There's a lot of wrapper functions out there. So so that you can understand it, I brought it down to like more bare minimums. So this here is we're finding the variance across the rows. Margin here is 1. So across the rows, we find the variance, and then we're sorting the genes based on the variance, and then we're taking the top 500 genes. So that's what this function here is. So we're taking the log counts matrix here because we're running Pca, and Pca tends to want log counts, even though logging the data distorts the data. And we're just finding the variance of the rows. So basically for every single gene, we're calculating its variance, we're sorting them, and then we're taking the first 500. Okay, so that's what this code here is doing. And if we run Pca on this, we don't really have to do the process time. And we're running pre-comp on this. So pre-comp here is a Pca method that we called earlier. We now have, oh, I shouldn't have done that. Let me just do structure instead. Oh, I didn't need to do that. Okay, let's run it twice. We now have basically the structure from pre-comp that we had already. We have our eigenvalues in the $SVD, and then we've got our dollar rotation here and our $X. Okay, we're just going to use this function plot embedding, and we're coloring here by cell type, and we can see here our Pca is not a very happy Pca because we've got this horrible horseshoe or arch in it. We can do, if we do additional pre-processing on the data, we do actually get rid of this arch. Okay. This here is plotting the dollar rotation. Okay. So the dollar rotation, as we mentioned before, is just the cell types. Just have a look what it looks like. These are basically the principal components of each of the cells. If we do, sorry, this is on the counts. If we run it on the log counts, as I was saying before, principal components analysis requires, oh, I didn't calculate it on the counts. if we run it on the log counts. This is just showing here the principal component analysis has to be done on the log counts, not the counts. And this here is just plotting the embeddings of the rotation. This here is just saying if you want to change which axes you want to see. And then the thing about it is, even if the principal components itself doesn't look great, this is on the log counts, it looks a little bit better. We normally find here on this principal component and the log counts here, we're beginning to see separation. There's less of that U effect, but that first component is still not giving us information. But even still, what we tend to do when we tend to do single cell analysis is we don't tend to look at the PCA plots because a lot of the times they look horrible. We just go straight to UMAP. And so UMAP or TISNE tend to be used as the second a part of any single cell RNA-seq analysis. We tend to do PCA and then UMAP or TISNE. And people tend not to plot the PCA, they tend to go straight to the TISNE or the UMAP. Now, TISNE and UMAP both have, both as part of them tend to use PCA at the start of them. In this case here, I'm calling UMAP directly from UMAP. From this function here, UMAP is within the package that I loaded already, or that UWAP package. When we run UMAP, we see this separation very easily. However, this is a really, really, really simple data set to separate out. If we want to get better decomposition and better representation of the data, if we give it better information at the start, you will do better. So these PCAs are not brilliant. If we actually simply run, and I'm just going to go down here to run correspondence analysis, and we don't have to look at the log counts here, we can just look at the standard counts matrix. We actually see much better separation from the start, and you actually then get better separation on the UMAP. So using correspondence analysis here is actually better than principal component analysis, or correspondence analysis is simply decomposition of the Pearson residuals. And it's actually very fast, as you could see there. I just ran that, it takes the same amount of time. Without doing anything fancy, this is just taking the counts matrix. I haven't done log counts, I haven't done anything fancy on it, just using the more appropriate decomposition. We're seeing much better results here on our single cell RNA-seq. And then of course we can still run UMAP on this. Plot embeddings, this is the log, this is the counts, so the log counts. And here we can run UMAP on it, and we can get a separation out here. One thing to note, if you're looking at UMAP or TISNE, the distances can sometimes be deceptive. We're not looking at a measured distance, particularly with TISNE. TISNE is an algorithm that tries to find the maximum separation by the clusters and plot them in the biggest space apart. A bigger separation space doesn't necessarily mean that you have real better separation. And you can see here, if we do the plot embedding, that we're actually seeing nice separation in the UMAP space. If we're using these values for downstream analysis, you're better taking from UMAP, definitely don't take from TISNE, or better still, use the actual values from the Corral or from the correspondence analysis. Those values you can take down into subsequent machine learning method, and you can actually interpret what those values mean. You can't really interpret the values from TISNE. So anyway, here we can begin to see that you've got some really nice separation in space between our 4 different cell types. Okay. And I think I'm coming up to time here. We have, I'm just going to see what questions we have. So I have a question about single cell. If I want to integrate multiple data sets, oh, sorry, I did actually, Fatima, I did answer that a little bit earlier. The other thing, so Corral here is within the package. Let me just go back to packages. It's within the package Corral. Within this, we also have a different function, this function corral M here, which is a multi-table alignment method. And that actually, you can call here either IRBLA, which is a fast version of SVD, you can call SVD here, and you give it either a list of matrices or one single cell experiment where you have splits in it. So from 1 to 100 is data set 1, from 100 to 400 is data set 2. This is one method. There's also within the Surrogate package, they have implemented canonical correlations analysis, CTE for aligning data sets as well that works together. There's another method called Harmony, which works. It uses Pca by default, but if you give it the corral methods, it works slightly better. And there's a couple of publications, I can definitely send them to Yago, but he can send them out to the group, where people have benchmarked different methods for integrating across batches. But definitely, if you start from the decomposition of the Pearson residuals, you'll actually do better. I think Yago, the materials and the lectures, they'll be available someplace, won't they?
Yes, we might send the link for them.
Okay, cool.
Yeah.
And the last thing I wanted to put up here was just at the end of the slides where I posted the slides. Just want to show one tab. Look at the slides. Oh no, I've just copied the wrong thing entirely. And now I open them twice. Which is going to win? Battle between the tabs. Okay. Please post questions there while I'm waiting for my slides to load. Is there a link for the handout? What do you mean the handout? Are you talking about the slides? Or are you talking about the vignettes? Okay, one person had information. How do I infer information from the plot? That's actually important. I will actually, if I haven't described that detail, I'll do very, very quick 1 second on this. But before I do that, oh my. Okay, maybe I will, while this is loading, I'll do the other question first. So if we're looking at a Pca plot, the first thing, this is a UMAP, whether it's a UMAP, whether it's a TITSNI, more so if it's principal components or any kind of dimension reduction, we normally plot the first axis on the horizontal. That's the first one. X is normally PC1 and Y is normally PC2. Now we may be looking at other principal components, but by default, the first one is on the x-axis. So the first thing I always look at is what's the separation on the x-axis. So when I'm looking at one of these plots, I look at the direction from the origin. So I say, well, what is most separated on the first component? Okay,